ELISA is a technique used for quantitative and qualitative estimation of antigen or antibody in a given serum sample. It is a fundamental test in modern biology. Some examples include detection of AIDS to measure soluble cytokine concentrations in tissue culture supernatant and to perform pregnancy tests. The major advantages of ELISA include it is highly sensitive and can detect even small amounts of antigen or antibodies in the sample. It follows a high throughput approach which allows multiple samples to be measured in a single experiment. This is made possible by the presence of 96 microtiter wells in the ELISA microplate. It can be easily automated as the absorbance of the colored product formed is measured using a microplate reader and data is stored in a computer. General procedure for ELISA is as follows. Coat the microtiter well with antigen or antibody. Wash it to remove unbound antigen or antibody. Combine the coated surface with the test material containing unknown amounts of antigen or antibody to form antigen-antibody complex. Wash to remove unbound antigen or antibody. Now link an enzyme with the antigen-antibody complex. For example, it can be done with the help of an enzyme-conjugated secondary antibody. Incubate it for some time and then wash. Add chromogenic substrate specific for enzyme and allow the reaction to occur. Now measure the absorbance of the colored product formed using microplate reader to quantify and detect the antigen or antibody concentration in the serum sample. Common enzymes used in ELISA are horseradish peroxidase, which converts its substrate TMB to a blue-colored product and its absorbance is measured at 615 nanometer. The second enzyme used is alkaline phosphatase, which converts its substrate PNPP to a yellow-colored product and its absorbance is measured at 405 nanometer. The third enzyme used is beta-galactosidase, which converts its substrate ONPG to a yellow-colored product and its absorbance is measured at 410 nanometer. Types of ELISA are direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. Direct ELISA. The procedure is as follows. Coat the microtiter well with antigen. Wash to remove unbound antigen. Now add antibody conjugated with enzyme and incubate. Incubation is necessary to allow the antibody to bind to the antigen. Wash to remove unbound antibodies. Now add the chromogenic substrate for enzyme and allow the reaction to occur. After this, measure the absorbance of the colored product formed using microplate reader. This type of ELISA is called direct ELISA because an antigen is immobilized in the well and an enzyme conjugated antibody directly binds to it. There is no need for secondary antibody conjugated with an enzyme. In case of direct ELISA, we measure the amount of antibody present in the serum sample. Here, absorbance is directly proportional to the amount of antibody present in the serum sample. Direct ELISA is a reliable technique for Mycoplasma bovis serological diagnosis. Indirect ELISA. The procedure is as follows. Coat the microtiter well with antigen. Wash to remove unbound antigen. Now add serum sample having unknown amount of antibody. This antibody is called primary antibody. Incubate it for some time to allow the binding of antibody to the antigen. Wash to remove the unbound primary antibodies. Now add enzyme conjugated secondary antibody and incubate to allow its binding to primary antibody. Wash to remove unbound secondary antibody. Add the chromogenic substrate for enzyme and allow the reaction to occur. After this, measure the absorbance of the colored product formed using microplate reader. This type of ELISA is called indirect because the enzyme which is causing the color reaction is conjugated with secondary antibody which is not directly bound to the immobilized antigen on the well. In case of indirect ELISA, we measure the amount of antibody present in the serum sample. Here, the absorbance is directly proportional to the amount of antibody present in the serum sample. Indirect ELISA is used in diagnostic tests for AIDS. Sandwich ELISA. The procedure is as follows. Coat the microtiter well with antibodies. Wash to remove unbound antibodies. Add serum sample having unknown amount of antigen and incubate to allow the antigen to get bound to the antibody. Wash to remove unbound antigens. Add a second enzyme, conjugated antibody, which is specific for a different epitope on antigen, and incubate. Wash to remove unbound second antibodies. Add the chromogenic substrate for the enzyme and allow the reaction to occur. After this, measure the absorbance of the colored product formed using microplate reader. This type of ELISA is called sandwich ELISA because it uses two antibodies with an antigen bound between them, somewhat like a sandwich. In sandwich ELISA, we measure the amount of antigen present in the serum sample. Here the absorbance is directly proportional to the amount of antigen present in the serum sample. Sandwich ELISA is useful for the measurement of soluble cytokine concentrations in tissue culture supernatant as well as in serum and body fluids. Competitive ELISA. The procedure is as follows. Incubate the antibody in the solution with serum sample containing unknown amount of antigen. Add the antigen-antibody mixture to the antigen-coated microtiter well and incubate. Incubation allows the unbound antibodies in the mixture to bind to the immobilized antigens on the well. Wash to remove unbound antibody. Add enzyme conjugated secondary antibody and incubate. Wash to remove unbound secondary antibodies. Add the chromogenic substrate for enzyme and allow the reaction to occur. After this, measure the absorbance of the colored product formed using microplate reader. This type of ELISA is called competitive ELISA because a competition occurs between the antigen of the serum sample and the antigen immobilized on the microtiter well to get bound to the primary antibody. In case of competitive ELISA, we measure the amount of antigen present in the serum sample. Here, the absorbance is inversely proportional to the amount of antigen present in the serum sample because the more the antigen present in the serum sample, the less free antibody will be available to bind to the antigen-coated microtiter well. Competitive ELISA are used in cases of heavy infection as during heavy infection, the antigen concentration in serum is very high. So in this case, when other methods of ELISA are used for detection and quantification of antigen, the intensity of colored product is so high that the absorbance measured by the microplate reader is inefficient. In such cases, competitive ELISA provides an accurate detection and quantification of the antigen since it is based on the principle that absorbance is inversely proportional to the amount of antigen present in the serum sample. These are the references which were helpful in making this video. Thank you.